Once you've selected sequences to add to your project from your BLAST search, the next step is what's called a muscle alignment. Muscle stands for multiple sequence comparison by log expectation. Essentially what a muscle alignment does is it takes all of the sequences that you've selected to add to your project and it aligns them or stacks them up on top of each other to compare them, and from there you can impute potential evolutionary relationships and relatedness between the sequences. So the first step in creating a muscle alignment is to add reference data that taxonomically match your samples and primers, but are not necessarily close BLAST hits. This allows you to select downstream what's called an outgroup, and I'll explain that to you in a little bit more detail very shortly. So to add reference data, what you're going to do is click the Add Reference Data, and then you'll have several choices of reference data to choose from. COI for animals, RBCL for plants, 16S for bacteria, and then ITS for fungi. So since we're analyzing fungi right now, we're going to select reference fungi from the ITS group. Once you've added your reference data, then you can select sequences to align. And this will essentially be all the BLAST hits that you selected from the previous activity, your samples in an individual outgroup. So this is how you'll do that. So this is going through right now, selecting all of your BLAST hits that you selected from the previous activity to add to the project. Then your consensus sequences of your unknown samples that you collected, and then an individual outgroup. And in this case, we're just going to select rust fungus, or sorry, smut fungus, and then hit the muscle button. And it'll take a few minutes to analyze. After about a minute, you'll get an output that looks like this. You're going to want to trim the ends off the alignment. So go ahead and click the Auto Trim button, and you should get a trimmed muscle alignment that looks similar to this. Here's your trimmed muscle alignment. Each row in this muscle alignment represents the DNA sequence of your samples or an individual hit from your BLAST search. The labels to the left of the alignment identify each sample. The numbers above the alignment at intervals of about 100 represent the base pair position in the sequence. The light gray areas in each column represent base pairs that match in each sequence. The colored areas represent polymorphisms, or locations where the base pairs are different across sequences. Each color is representative of a specific nucleotide. A's are green, T's are red, C's are blues, G's are black. The top row that says sequence conservation represents how often there are differences in the nucleotides at that position across all the samples you're comparing. The more white you see, the more often we see a nucleotide that is different from the most common nucleotide at that position. The second row down that says sequence variation shows you all the different nucleotides that are present at that position in the sequence across all the samples that are being compared. The third row down, labeled consensus, shows you the most common nucleotide at each position in the sequence. For instance, if you align 5 sequences and the nucleotides at position 20 are A, T, A, and G, then the consensus sequence should have an A at position 20 because A is the most common nucleotide at that position. All sequences are unknown, reference, and close matches are being compared to each other. The consensus row doesn't really need to be understood in detail, but it represents the nucleotide that matches in all sequences where a polymorphism or colored line is not shown. Lastly, dark gray areas, if you see them in your alignment, represent gaps or spots where the sequence is missing from a particular sequence in the alignment. These gaps can be created by the insertion or deletion of large chunks of DNA in some but not all of the species being compared. If you hit the ATCG blue button in the upper right hand corner, it'll zoom in on the muscle alignment so you can actually see the individual nucleotides labeled by the letter. So go ahead and click that in your DNA Subway screen and then answer these questions. Go ahead and hit pause until you've got the answers. Okay, so what is the maximum number of polymorphisms possible at any one site in the barcode? Well, the maximum possible number is, of course, 4. This is the maximum number of individual nucleotides that can be at any one site. It can either be A, T, G, or C. What is the most common base across all the sequences at site 5 is indicated by the arrow in the upper left corner there, site 5. The answer is A, as indicated by the consensus sequence. A is the most common letter if you compare what letter is at that point across all of the sequences being compared. And what does tray, or sample number 4, have at site 5? The answer indicated by what's in that blue square is once again A. And that's because tray doesn't show a letter there because he matches the consensus nucleotide there, which once again is A. If you don't see a letter there, that means tray, or whatever the sample is, has the most common nucleotide, or the same as what's listed in the consensus sequence. You're likely asking yourself, okay, so what do I actually need to pull out of this muscle alignment? The two big questions that you'll need to answer from this muscle alignment are, number 2, which of all the taxa that are lined up is most similar to your particular unknowns? So in this example, let's say, which of these is most similar to Trey's unknown? And you'll also need to identify which taxon or which individual sort of sequence line is least similar to all of the unknowns. So if we look at Trey's sample, it's tough to say which one is the most similar, whether it's Hymenopellis incognita, Limnospora, Megalospora, radicata, or orientalis. Likewise, it's very difficult to say which taxon is the least similar across all of our unknowns. So a really useful tool to help tease this apart in a quantifiable way is the sequence similarity table. So let's look at that on the next slide. So once you click on the sequence similarity table, you should see something like this. This is a % sequence similarity matrix. It's a table that does a cross comparison of every single sequence that you're looking at in your muscle alignment. Sequences labeled 1 through 24. So for example, we know that Trey's sample is sample number 4. And so we can look at the highlighted area here and follow that row the whole way across to see which % sequence similarity is the highest between Trey's sample and the samples that we're comparing him to. And what you can see is 96.77 is the highest number. So samples number 5 and 6 are 96.77% similar to Trey's sample. They have the highest % sequence similarity in that muscle alignment. And so those samples, Hymenopellis, are both Hymenopellis limnospora. And so they have the highest sequence similarity to Trey's sample. We can also begin to decipher which taxon is the least similar to all of our unknowns. Can you hit pause and figure it out on your own? If you said sample 1, you're correct. If you look at the % similarity of sample 1 to all the other samples, you'll find that sample 1, the SMUT, is the least similar, about 55 to 67% similar across all our groups. So sample 1 will be our out group. And apologies that it says Baker's yeast there, it should say SMUT. SMUT will be the out group.